The other possible method, and this is the one we're going to be focusing on in this session, of course we've already heard a little bit about this in the previous session, and that's editing the egg or zygote, fertilized egg or at fertilization. In this case, as George pointed out, we have this problem of how do you verify the edits? How do you know that you got what you wanted and no off-target events? Pre-implantation genetic diagnosis, in theory, could be used, but for that to work, you have to know that the embryo is not mosaic. If there's a mixture of different genotypes in your cells in the early embryo, then you'll never know whether you've taken one with an edit or one without an edit. You won't know what's actually happened in the rest of the embryo. However, there are several new methods reported to allow very high efficiencies, and they could perhaps approach 100%, which is what you'd need to have some guarantee that the methods are really going to work. Especially if those methods are combined together, I think that there's a prospect of getting really high efficiencies. We also need, though, to know a little bit more about what's going on in the early embryo, particularly with DNA repair mechanisms, for example. So broadly, these new methods involve careful choice of genome editing components, ways to make them more efficient. For example, ways to, if you're going to use homology-directed repair and have a template, ways of bringing the DNA template to the Cas9 guide RNA complex. And then also choosing the right stage in the early embryo when you should do this modification, because that may be critical. Right, so without any further talk from me, I want to invite Kathy Nayakan, who's at the Francis Crick Institute in London, to present her talk, which is going to be basically focused around using genome editing to ask questions about the biology of early human embryos, and also in the process what you learn about genome editing. Thank you.
So I want to begin by thanking the organizers of the summit and for Robin for the introduction. I'm really thrilled to be here. So I wanted to start by describing some of the interests that my lab has in understanding early human development. Currently, we know very little about how the earliest cell types in a human embryo are specialized or become specified in their fate and function, and what are the key genes that are responsible for this process. So we're using CRISPR-Cas9 methods of genome editing, among many other methods, to understand and address broader questions about gene function in early human embryogenesis. So I wanted to illustrate this in a figure of a human embryo, which has been stained to look at the expression of 3 key proteins that distinguish the 3 different cell types that are comprised in this human embryo just before it implants in the womb. So at this stage of development, the human embryo is comprised of approximately 200 cells and is technically called a blastocyst. And it's only a small subset of cells, these cells marked here in green, that express a key protein called OCT4, which I'll talk quite a lot about. And it's only these OCT4 expressing so-called epiblast progenitor cells that have the unique potential to give rise to the entire embryo proper or the fetus. But at this stage, there are approximately 10 out of the 200 or so cells express OCT4 and are fated to form the entire embryo. The majority of cells marked here in blue are precursor cells of the placenta, and the third cell type that's comprised of within the human embryo are marking and distinguish yolk sac progenitor cells. So we've been studying mechanisms of how the first cell types in a human embryo become specialized in human development immediately after the egg is fertilized all the way up until the human embryo forms this so-called blastocyst, approximately a week after fertilization. And the reason why we study these mechanisms and processes in human embryos is that we'd like to understand fundamental aspects of our own biology. And work that I'll show you shortly suggests that there are fundamental differences when comparing early human embryogenesis to how embryos develop in other organisms. The other reason why we study these developmental events directly in human embryos is it has profound clinical importance in vitro fertilization therapies. So most human IVF embryos fail to reach this blastocyst stage. And of the embryos that do develop to form a blastocyst, 50% will fail to implant. And of the embryos that do implant, approximately 50% will fail to develop beyond 3 months. So these are success rates of approximately 12 or 13% that are really worldwide rates of in vitro fertilization treatments. So we think that by better understanding how early human embryos develop to form a blastocyst and potentially identify molecular criteria that are better predictors of implantation, we could potentially improve on these rates. Because the current methods that are used in IVF clinics utilize, for example, just gross morphological features of the embryo and no molecular criteria whatsoever. The third reason why we're interested in studying these early developmental events in human embryos is it has tremendous importance for embryonic stem cell biology and regenerative medicine. So it's from those OCT4 expressing epiblast progenitor cells that we can also coax these cells to self-renew indefinitely as embryonic stem cells. So they make more copies of themselves, they replicate, and they can also be directed to differentiate into any cell type, theoretically, in the human body. So we can use these embryonic stem cells to inform our understanding of disease progression. So for example, by generating neurons to understand neurodegenerative diseases. Theoretically, these embryonic stem cells could potentially make therapeutically relevant cell types for cell replacement therapies. And importantly, these cells could also be used to screen for drugs that might potentially ameliorate disease phenotypes. But fundamentally, embryonic stem cells and their derivation is a rather inefficient process, and most human embryos fail to give rise to embryonic stem cell lines. Moreover, we know that the process of generating induced pluripotent stem cells is also inefficient, and quite an active area of research in the stem cell field is trying to understand why human embryonic stem cells derived in vitro in a petri dish are molecularly distinct or different from those early embryo cell types from which they were derived. So the process of in vitro culture of these cells somehow allows these cells to acquire molecular properties that are distinct, and we know that established human embryonic stem cell lines are not equivalent in their differentiation potential. So there's quite a lot of additional work that's needed in the stem cell field to kind of better establish cellular models of these early cell types. The other reason I'm showing this slide is also to highlight that we and many others for a number of years have been using model organisms like rodents, the mouse, to understand human biology, and that's because at these early pre-implantation stages that we focus on, the gross morphology of the embryos are quite similar, but we know that there are important distinctions between our species. And one example is the timing of when there's active transcription in of the genomes, which occurs much earlier in the mouse compared to the human. And there are differences in the timing of when embryos implant, and then after implantation, there are obvious molecular and morphological distinctions. So we've invested in transcriptionally profiling and identifying all of the key genes that are expressed in human embryos throughout these early pre-implantation stages at the single cell level, and comparing the gene expression dynamics in human embryos to the mouse. And what we found is that for the vast majority of genes, homologous or potentially orthologous genes, there are differences in the timing of gene expression. So these are two of the several thousands of examples I could show you So here we have plotted developmental time on the x-axis and gene expression on the y-axis, and we can detect the transcript for a gene called POW5F1. This encodes that key factor I mentioned earlier, OCT4. POW5F1 transcripts are detectable much earlier in the mouse compared to the human, and another absolutely critical developmental regulator called KLF4 is also detectable much earlier in the mouse compared to humans. And in addition to temporal differences in gene expression between our species, we also see differences in cell type specific expression. So again, these are just a few of the thousands of examples I could show you. So here we're just looking at this so-called blastocyst stage just before implantation, and we can identify transcripts of factors that are critical regulators of the so-called pluripotent compartment in a developing mouse embryo that are absolutely also required for establishing mouse embryonic stem cells, and they are, as one would predict, enriched in the pluripotent compartment in the mouse, but interestingly are undetectable in the human at these early stages. And conversely, we see factors which are highly enriched in the pluripotent epiblast progenitor cells, these green cells, which are undetectable in the mouse. And so we'd like to study the function of these genes. We predict some of these genes might have really important roles for the development of the human epiblast, but we can't use kind of this particular model organism to understand the biology. And indeed, some of these factors are no longer expressed in established human embryonic stem cell lines, so they're lost in vitro cultured cell lines. So for these reasons, we'd like to explore gene function directly in human embryos. And we decided to target a gene, the gene OCT4, or the protein OCT4, directly in human embryos as a so-called proof of principle, as a positive control, because we predicted that OCT4 might have a really critical role in human embryogenesis, given its important role in the mouse before implantation. And the other reason why we selected OCT4 is that we showed previously OCT4 protein is detectable from as early as around the 8-cell stage in human embryos, and its expression is critically required for the establishment of embryonic stem cells, both in humans and in mice. So to identify methodologies to target OCT4, we used a number of different cellular systems to optimize techniques. And here we optimized methods to target OCT4 and screen various different guide RNAs in human embryonic stem cells, where we could flip in various different guide RNAs and test their relative efficiencies in these in this isogenic background. So we could, in the presence of tetracycline, screen for guide RNAs that induce the loss of OCT4 protein in the background of constitutive or constant expression of this Cas9 enzyme. So to make a very long story short, we identified a guide RNA boxed here that in the presence of tetracycline induced the highest proportion of embryonic stem cells that had detectable insertion-deletion mutations. And when we interrogated the types of indel mutations observed when we induced the expression of this particular guide RNA, we saw that the most frequent type of indel mutation was this 2 base pair deletion, suggesting the potential mechanism for the repair of the double-strand break induced might be microhomology-mediated unjoining. We also invested in optimizing methodologies to introduce the CRISPR-Cas9 components into mammalian cells, and we screened this via mouse zygote microinjections. And what we identified is that by microinjecting the ribonucleoprotein complex of Cas9 together with our guide RNA, we saw the highest proportion of OCT4 null mutant mouse embryos, and we saw a reduced mutational spectrum, suggesting that the microinjection of the CRISPR-Cas9 components into zygotes resolved in indel mutations at the 2-cell stage or earlier in the majority of mouse embryos. So with those methodologies, we then microinjected the CRISPR-Cas9 component as a ribonucleoprotein complex directly into human embryos. So these are embryos that are surplus. They're spare to in vitro fertility treatment. And many of the couples had gone to tremendous lengths to donate their surplus embryos to our research project. And these embryos were frozen as 2 pronuclear stage zygotes, so we can see maternal and paternal pronuclei. They're packaged in distinct nuclear envelopes. Here, the human zygote is surrounded by a shell called the zona pellucida, and we're microinjecting the CRISPR-Cas9 components into both pronuclei. We then cultured these embryos and compared the development of control-injected embryos to embryos where we're targeting OCT4, and I'm showing these videos asynchronously. So in both cases, the embryos divide, they cleave, they make 2 cells, 4 cells, 8 cells. They undergo a process called compaction here, and then they initiate this formation of the blastocyst. So you see a blastocele cavity here and here. The embryo starts to expand, but in comparison to the control, the OCT4 targeted embryos underwent iterative rounds of expanding and collapsing and eventually arrested. The embryos failed to hatch from this shell called the zona pellucida, unlike this control-injected embryo where we see some partial hatching here, and that's an absolutely critical process in IVF embryos to allow for implantation. When we compared the rates of cleavage division, there were no significant differences, but significantly fewer OCT4 targeted embryos developed to this blastocyst stage, suggesting a role for OCT4 in early human embryogenesis. We then wanted to phenotype and genotype these embryos, and we were predicting quite a large degree of mosaicism. So we invested in developing methodologies to extract from a single cell both the genomic DNA for genotyping and karyotype analysis, and as well as the RNA from the same cell to perform transcriptomics analysis to phenotype if there are any changes in global gene expression. And here you're seeing in the filled-in shapes, the OCT4 targeted embryos are transcriptionally distinct from the unedited embryos. And a number of key developmental regulators were differentially expressed, and I'll just show you one example of what we saw. So in the mouse, OCT4 null mutant mouse embryos retain the expression of placental progenitor markers. And indeed, these placental markers are also ectopically expressed in these embryos, suggesting that OCT4 functions as a negative regulator of the placental program in the mouse. And in fact, when we isolate these mouse OCT4 null embryos and plate them in conditions that ought to give rise to mouse embryonic stem cells, or these inner cell mouse outgrowths, they predominantly give rise to placental outgrowths called the trophoblast. But in the human, we see kind of the diametrically opposite effect. So in human embryos where we've targeted OCT4, despite quite a high degree of mosaicism, we see the reduction of placental markers like GATA2, CDX2, which I'm not showing, and dysregulation of markers that distinguish tight junctions between the placenta, which are absolutely critically required for the expansion of the human blastocyst and may explain why the embryos went through these cycles of expanding and collapsing. So just in the last 2 slides, I wanted to talk about some of the caveats of the work. So one of the things that concerned us is whether at the on-target site, if we were accurately capturing the targeting events that occurred at these sites, or whether they may also encompass more complex mutations. So for this, we've been walking along this POW5F1 gene locus and performing single nucleotide polymorphism analysis. And what we found is that in some of the cells isolated from an embryo, we can detect a single nucleotide polymorphism in close proximity to the on-target mutation. So here, a 2 base pair and 3 base pair deletion within a single cell, we can detect a SNP at this site. But when we isolate a single cell from the exact same embryo where we would expect to see and detect both alleles, we see allele dropout. So now we've lost one of the SNPs. So we're only able to PCR amplify one of the parental alleles. we think. And what we wonder is if we SNP type along this locus, if we reestablish a SNP and bookend the sites of what we think might be a large deletion, whether we can amplify across this SNP and determine exactly what's going on and how the double-strand break was repaired at this on-target site. In addition, we've been performing whole genome DNA sequencing on single cells isolated from the OCT4 targeted embryos. And in some of the OCT4 targeted embryos, we're able to detect 2 copies of chromosome 6, which is where the PAL5F1 or the OCT4 locus is. However, in other embryos, we could detect some cells where there's a segmental loss of chromosome 6 that encompasses approximately 29 megabase pairs of genomic DNA sequence. And in other embryos, we see segmental gains in close proximity to where the double-strand break would have been induced. So I think this is a significant note of caution and may suggest possible complexities at the on-target site that need to be considered in both basic research and also thinking about potential clinical applications. So just in summary and in terms of our future aims, we're interested in understanding early human development. We're using novel technologies such as CRISPR-Cas9 to understand gene function in human embryos. We'd like to use this information ultimately to identify potential biomarkers of successfully developing human embryos and improve IVF culture conditions. And we'd also like to use this knowledge to define requirements for human-induced pluripotent stem cells and human embryonic stem cells. So I'd like to thank members of the lab, and in particular our clinical collaborators at Bourne Hall Clinic and our funders. Thank you very much.
Thank you very much, Kathy. So our next speaker is Paula Amato, who's at the Oregon Health and Science University, who's going to be talking about, I guess, homology-direct repair, or particularly interhomolog repair in early human embryos.
Thank you. Thank you, Robin, for the introduction, and thank you to the organizers for inviting me today. I'm pleased to be here to share with you our work from the Metalopov lab on human germline gene therapy. I have no relevant financial conflicts of interest. I'd like to update you on 2 of our projects. I want to briefly mention mitochondrial replacement therapy. Although it doesn't involve gene editing per se, it is a form of germline gene therapy, in fact, the first form of germline gene therapy. And I think some of the issues are similar, so are worth mentioning. And then, of course, I'd like to talk about our work on nuclear gene editing or gene correction. So currently, if a patient or couple is afflicted by a genetic disease, they have a few available strategies to try and prevent transmission of that disease to their children. They could certainly pursue adoption, or gamete donation or embryo donation, or they can undergo in vitro fertilization with PGD, which essentially is a method of embryo selection, but perhaps in future they could avail themselves as well of germline gene therapy. And this, of course, involves correcting the disease-causing gene mutation in the reproductive cells themselves, either the sperm, the egg, or the embryo. Of course, this is much more controversial because these changes in the germline are heritable to all future generations. So just a reminder, our mitochondrial DNA is separate from our nuclear DNA. We inherit our mitochondrial DNA exclusively from our mothers. And whether or not somebody exhibits mitochondrial disease really depends on the degree of heteroplasmy, the ratio of mutant DNA to wild-type DNA. And this can be variable within children in the same family, and even in the same person in different tissues, there can be different degrees of heteroplasmy, making PGD actually very challenging for these cases. So several years ago, Dr. Metalopov proposed a method to circumvent mitochondrial disease, and it's called spindle transfer. It involves taking the nuclear DNA from an egg from a woman who's affected with a mitochondrial gene mutation, and then transferring that nuclear DNA into another donor egg that's had its nuclear DNA removed, and then fertilizing that reconstituted egg, making an embryo and hopefully eventually a baby, the so-called three-parent baby, which is a bit of a misnomer, but point is the DNA would come from 3 different individuals. And he did this initially in a monkey model using 2 different species of monkeys, and it was successful. And these monkeys have been followed now for several years, and they appear to be healthy with low levels of heteroplasmy. In fact, we have 2 generations of these monkeys, and they appear to be healthy. We then moved this work into the human model in vitro, and we recruited healthy young egg donors, and similarly took the spindle out of one egg and transferred it into another egg that's had its spindle removed. We fertilized those eggs with donor sperm and created embryos, and we were able to show that these embryos were developmentally normal and that the carryover of the original mitochondrial DNA was less than 1%. We initially did this in healthy young women, and then we recruited women who actually carried a mitochondrial gene mutation, repeated the same study, and found similar results. In some cell lines, we did observe reversion back to the original mitochondrial DNA haplotype, about 15% of cases. We're not sure yet why this happens. We think that certain mitochondrial haplotypes might have a replicative advantage, but certainly it is important in terms of counseling patients, if this were to move to the clinic, that this is a possibility. It may be that haplotype matching, similar to what we do for organ transplantation, might be necessary. So to summarize this part of the talk, mitochondrial replacement therapy is applicable to any mitochondrial gene mutation because you're essentially replacing the entire mitochondrial genome. We have now evidence from preclinical studies in animals and in vitro in humans that it appears to be safe and effective. We do have this reversion phenomenon that we've observed that's important. As I mentioned, haplotype matching may be required. And clinical trials are currently ongoing in the UK. Preclinical studies are continuing in the US, and there have been a few babies born from this technique already, notably a baby in Mexico a couple of years ago, and a few other babies have been reported born in Ukraine. So we then switched our attention to nuclear gene mutations. The development of CRISPR-Cas9 allowed us to target nuclear genes, and the first gene we targeted was one called MYBPC3. This is an autosomal dominant mutation that causes a condition called hypertrophic cardiomyopathy, which leads to an enlarged heart, which can cause sudden cardiac death in people who have this condition. So we recruited a gentleman who had this mutation. So his sperm, he's heterozygous, his sperm, half his sperm are wild type and half are mutant. We designed a CRISPR-Cas9 complex to target this particular mutation and provided an external DNA template for the embryo to use to repair itself. We again recruited young, healthy women to donate eggs, fertilize these eggs with this gentleman's sperm, and then analyze the embryos at the 8th cell stage. And what we observed was a higher percentage of wild-type embryos than expected. You would have expected 50-50, and we observed closer to 75% of the embryos were wild-type, suggesting that there was indeed some repair happening. Initially, we injected the CRISPR-Cas9 complex at the zygote stage, the 1-cell stage, but we found that when we did that, there was a significant amount of mosaicism, which means some of the cells were corrected and others were not. We then switched and started injecting the CRISPR-Cas9 at the same time that we injected the sperm at the time of ICSI, and we found that we significantly reduced the degree of mosaicism. These embryos, again, were developmentally normal and karyotypically normal. We did look for off-target effects. We used the DiGene platform developed by one of our collaborators, Jin-Soo Kim, and we did not observe any off-target effects in our experiments. Interestingly, when we looked at the mechanism of repair, we were surprised to see that the embryo did not use the external DNA template that we had provided, yet we were seeing this repair, so we hypothesized that the embryo must have been using the wild-type allele that came from the egg, in this case, to repair itself. This phenomenon called gene conversion, it's been described in other species and certainly other cell types, somatic cells, but had not been described in the embryo before. Shortly after we published our work, there was some skepticism about our proposed mechanism, and people wondered whether we had missed a large deletion that allowed us to fail to detect the mutant allele. So we did additional studies, including long-range PCR up to 10,000 kb, looking for large deletions, and we didn't observe any large deletions, so we think our original hypothesis is correct. When we looked at the gene conversion more closely, we actually saw that the conversion extended both upstream and downstream from the actual mutation site. And we could see this because we used informative SNPs from the sperm donor and the egg donor. And this raises new safety concerns now, because you can imagine if you have loss of heterozygosity in genes adjacent to the gene of interest, you could either inactivate genes that are important or activate oncogenes. So this is something to be aware of. We have since tried this with other gene mutations. Heterozygous gene mutation have seen the exact same results. We tried this with homozygous mutation, and the embryo again did not want to use the external DNA template that we provided. The efficiency of HDR repair was very low, less than 5%, certainly would not be applicable for clinical purposes. Most of the repair happened by non-homologous end joining, which of course would not be desirable in a clinical setting. So to summarize this part, we believe, at least for heterozygous mutations, that germline gene editing is highly efficacious and efficient. The results have now been replicated by other laboratories, and we think that this will serve as a complement to PGD, and I'll say more about that in a second. We think it does prevent some advantages over treatment, as was alluded to in the previous session. If you can prevent disease simply from a healthcare cost savings point of view, it's a lot cheaper to prevent a disease than have to treat it once it occurs. But we strongly believe that the efficiency, safety, and ethical concerns need to be addressed before moving this work to clinical trials. I just want to briefly mention again the report by the NIS published in 2017, which set out a set of criteria for germline gene editing. And the very first one is absence of reasonable alternatives. And people always point to PGD. Well, why do you even have to do this? You can do PGD in most cases. And I would like to suggest that this criteria, at least the first one, be revisited, because I think if a technique offers some advantage, significant advantage over an existing alternative, then there's justification to pursue it. So this is a series from a group of clinics, fertility clinics in France, and looking at their experience with IVF and PGD over a 5-year period. So there were over 300 couples that were doing PGD for various genetic diseases. They underwent over 500 cycles of IVF, egg retrievals. 3,000 embryos were generated. Of those, just over 1,000 of them were unaffected. 600 were transferred, giving you a delivery rate of about 18%, so 95 deliveries to over 500 plus thousand cycles. But interestingly, there's another 1,000 or so embryos there that are affected with the mutation, and at least 700 of those were otherwise morphologically normal. So if this germline gene editing is shown ultimately to be safe, why wouldn't we want to correct the mutation in those embryos and increase the efficiency of this PGD process, reduce the burden and cost of IVF? As far as our future plans and directions, we're pursuing MRT trials outside of the United States. We're looking to use MRT for other indications such as age-related infertility. We're continuing our work on germline gene editing, looking at other genes, including BRCA and Huntington's disease. And we believe that the insights that we're learning about DNA repair from human embryos is going to be clinically applicable to somatic gene therapy for other conditions in medicine, such as aging and cancer. And finally, I'd like to thank all the people involved in the work, specifically Dr. Shukrat Matalipov, who's led this work for many years, all the members of his laboratory, my colleagues at OHSU, and our collaborators around the world, and to acknowledge the funding from OHSU. Thank you very much.
Thank you very much, Paula. So our next speaker is Maria Jason from Memorial Sloan Kettering Cancer Institute, New York. Okay. She is going to talk about the basically the peculiar development of the early human embryo or early embryo first cell cycle, for example, and its relevance of that to the genome editing. Sorry, we have a slight technical problem.
Yes. The PowerPoint is not responding. What's wrong with the PowerPoint? It is not responding.
Okay, thank you for rescuing the presentation. So I'm pleased to be here, and I hope I'm a good follow-up to the last two talks. It's more of a basic presentation to describe some of the complexities we have to consider, and I think until some of these recent studies, like from the Matalipov lab, it wasn't so apparent how much we don't know. So I'm going to, as Robin mentioned, discuss 2 points. One is the unique features of the first embryonic cell cycle, and then the other is, especially near and dear to my heart, the complexities of double-strand break repair. This is the start of a human embryo. In this case, it's an injection of sperm, but you get a fertilized egg. And I think one of the things that people often don't really think about if we're used to mammalian cell culture and gene editing there, that this embryo starts with 2 copies of the maternal genome, which will then have to still divide, and a sperm genome that has a very different chromatin structure compared to a normal nuclear genome. So it comes in pretty deficient in histones and is instead highly compact in protamine proteins that have to be completely replaced. And then Soon after, there's the division of the maternal genome, so this second meiotic division, while the paternal genome is undergoing this massive chromatin reorganization. And then finally, this very unique stage, as far as I know, where there are two nuclei called pronuclei with the maternal and paternal genomes. So as was just mentioned, the advantage to very early injections of Cas9 is that the maternal and paternal genomes at this stage are a single copy, and so this would mean that immediate gene editing would lead to embryos without mosaicism, which is highly preferred. But there are complications because the two genomes are so different chromatin-wise. The paternal genome seems to be much more proficient in gene editing than the maternal genome, at least in a mouse study that's been published. And then the other point, again, is that the maternal genome is undergoing this second meiotic division, and so one would think at this point that interactions between the two genomes before the pronuclei have formed would be discouraged. The other option is to do Cas9 injections later, and this would be about the time of DNA replication. Now the issue is that each genome is going to be 2N, and so this can lead to a high frequency of mosaicism that, again, was just mentioned in human embryos, and those of us working in mouse embryos are very aware of this, and that it's very difficult to get out clean genotypes in injected eggs. So the individual then has two different cell types in its body, one that might be edited, say, and the other one that's still unedited. We think of this as very disadvantageous especially in the human genome editing field, because one would want an individual with a very defined genetic change, although actually in thinking about the recent CCR5 case and CCR5 having a utility in some infections but not in others, maybe it actually would assist that individual in being not infectable by HIV, but able to respond to other viruses. There is another aspect that's quite unique. So the maternal and paternal genomes, again, after are in separate pronuclei and then undergo nuclear envelope breakdown, and it actually takes a while for the two genomes to really be integrated the way they normally are in our nuclei. So one can take out the two genomes separately. And in fact, the Ellenberg lab just recently published how separate the genomes are even upon the first division because there are two separate spindles that are formed, and maybe this is one of the reasons why human embryos are so poor in terms of having a normal chromosome complement. So I now want to switch to double-strand break repair. And for more than 20 years, we've worked with this paradigm for gene editing that a double-strand break will induce homologous recombination, but also non-homologous end joining, leading to mutagenesis, so precise change and imprecise change that can knock out genes. And one can see this in a single experiment where non-homologous end joining events are seen, as well as homologous recombination events, although usually non-homologous end joining is more efficient. And this is an experiment after the transfection of a nuclease, and you can see products within 4 hours after transfection, and it looks like the NHEJ events are a little faster than the HR events. Now, these are the two pathways we focus on the most, but there are additional repair pathways that we know about. Another version of non-homologous end joining, but also this single-strand annealing pathway where there can be deletions that are formed. But even the homologous recombination pathway is not a single pathway, and this is from a review I did with Rod Rothstein, and you can see the complexity and the different types of events that occur and sometimes the outcomes. And each of these pathways involves different components. And then gene editing with single-strand donors, we don't know of a physiological role for that kind of event, but that is additionally distinct from some of these. And besides the standard smaller events, are the ones that lead to gross chromosomal rearrangements. So in this case, chromosomal translocations, but other rearrangements are possible as well, including deletions, large deletions, insertions, chromosome fusions, et cetera. So in considering the embryo and the changes that one would want to make, it's really imperative to be able to rule out any of these more gross events. So another thing to consider is that there are multiple homologous recombination templates in cells. Sister chromatids, this is after DNA replication, so sister chromatids, homologous chromosomes, and then sequence repeats on other chromosomes or on the same chromosome. This, to some extent, is disfavored because repeats on like ALU repeats or other kinds of repetitive elements are often quite diverged from each other. So we mostly focus on intersister recombination and sometimes interhomolog recombination. And intersister recombination is what we think is the predominant pathway, which I think is interesting to note in terms of nuclease approaches, because both copies of the sister can be broken at the same time, ruling out intersister repair in those situations. But the normal role of recombination, we think, is a little different from the kind of editing events we do, where there's a single-ended double strand break that then has to reinvade to restart replication. And so homologous recombination is actually absolutely critical to cells, and so homologous recombination deficient cells can be inviable, but they can also show numerous genome rearrangements. Now, in any kind of diploid cell, recombination between homologs can also occur, but in most experimental settings, this has been shown to be much less frequent than intersister events, although there have not been really very many studies. And while they can also be relatively kind of minor in terms of genome effects, they can also lead to crossover events. And this is extremely deleterious if it involves changes that, for example, are going to uncover a tumor suppressor gene. And so it would seem that this would be disfavored, and I think this is important to understand if this happens in the embryo. But interhomolog recombination is absolutely critical in germ cell development for making sperm and eggs, and that's because of its mechanical role for segregating homologous chromosomes. And so in the absence of recombination, then the chromosomes can never congress at the metaphase plate to accurately segregate. But one thing to note about this process is that it occurs through the programmed introduction of hundreds of double-strand breaks. So the cell is actually able to deal with a very large DNA damage load. But again, this is a very controlled process. And then finally, if we consider homolog recombination, we have to consider how the events lead to loss of heterozygosity, and usually crossovers have a larger gene conversion tract than non-crossovers, which in meiosis generally involves 50 base pairs or less. And so again, there needs to be very high confidence in any genotyping that's done in embryos to rule out deletions within a chromosome or loss of chromosome arms, as Kathy just showed, or even grosser events like translocations. Okay, thank you.
Okay, thank you. Can we have our next speaker? Thank you. Who is Jingzhu Huang from Shanghai Tech University, who is going to tell us about his experience of genome editing in early human embryos, I guess particularly using base editing.
So good morning, everyone. So it's most pleasure to be here. So and get nervous to see so many people around. So my lab has long been working with genome targeting in vivo. Then, as we know, CRISPR came out as a very powerful genome editing tools. So we first perform in vivo CRISPR genomic editing in mice. Later on, successful in vivo genome editing in monkeys, shown here. But very quickly, people's attention, lots of off-target activity was detected in vitro. And meanwhile, we perform comprehensive analysis in vivo and do find some like off-target activity, like shown here in mouse, in rat, in goat. And once Junzhou first demonstrated the successful targeting by CRISPR in human discarded tripronuclear zygotes. With this, meanwhile, it raised concerns about the efficacy, safety, and modality of the CRISPR. So then people tested lots of different kind of strategy, try to improve the specificity of CRISPR. And later on, more CRISPR genome editing in human embryo have been published and reported by my previous speaker. And with time, and more like a professional and also the public people realize the human genome editing for the therapeutic purpose is acceptable. We have been working with the dual sRNA Cas9 system to minimize the off-target mutagenesis, and the first success in vitro by use different kind of cell line. And later on, we demonstrated the versatility of the strategy in vivo, here shown in mice. And by whole genome sequencing analysis, we demonstrate the layer of target are still observed. And we also try the strategy in the discarded human tripronuclear embryo, and demonstrate it works well to decrease the off-target. And meanwhile, we find the off-target induced by CRISPR-Cas9 actually was affected by deaminase appleback. It was introduced by Dr. David Liu yesterday, and take the advantage of this deamination. And another evaluated deaminase. Dr. Liu's lab developed the CBE and the ABE, both perform programmable editing of a target base in genome by deamination without a double-strand break. Then it demonstrated it can decrease the off-target dramatically. And then we, based on his group's work, we try to develop in vivo basically toolbox. We first use another Cas9 CPF1 and demonstrated a high fidelity CT-based editing in vitro, in cell line. Meanwhile, we replace the appleback with another deaminase, A3A, which perform comprehensive CT-based editing in cell line also. We also develop BE+ system, which taking the advantage of some tag to take multiple copy of the deaminase to the target region to increase the editing scope. And it also enhanced the fidelity of CT-based editing, shown here dramatically decreased indel and unwanted C to A or G conversion. With these tools, then we performed BE-mediated CT gene editing in vivo in mouse here. Then perform analysis by deep sequencing to show the no off-target was detectable here. And meanwhile, use ABE to mediate efficient AG-based editing in vivo here by model the AR and Hoxstate 13 pathogenic mutation. And with this mutation, the mouse to recapitulate the disorder of here shown is AR insensitive syndrome, and another one is fused digit, and very good to see no off-target was detected by using whole genome sequencing combined with deep sequencing. So with all the in vivo data, we believe base editor can pinpoint the gene editing in vivo, and suggesting base editing could be a safe and efficient in vivo genome editing approach. Then this data inspire us to try efficient and pure in vivo CT conversion by use here shown CBE, which efficiently perform the base editing in discarded human trinuclear oocytes, but still we can detect very rare off-target by CBE. Meanwhile, we perform the efficient pure in vivo AG conversion by ABE, and conventionally no off-target was detected by deep sequencing combined with whole genome sequencing. We also demonstrated the base editor mediated isogenic genome editing in vivo, shown here by genotyping the blood smear to see only one kind of genotype was detected. And so with this success, we want to test the collection of pathogenic mutation of genetic disease, as mentioned by previous speaker. There are an estimated 6,000 to 8,000 rare diseases, and the rare disease are estimated to collectively affect more than 200 million people worldwide, and 3/4 affect the children, and over half are life-limiting, and over 80% of rare diseases are genetic in origin. Most are caused by defects in a single gene. Also, both the central nervous system and peripheral organs and tissues are affected in most rare diseases, and there are treatment for only 6% and even less than 6% of rare diseases, of which fewer than 1% are curative. So, and also it was too expensive, and the average of $1 million per treatment. So to this end, we choose Marfan syndrome pathogenic mutation here, and we first tested the hypothesis by generating a mutant cell line, then cure the mutation by base editor in cell line. After that, we use discussed the tripronuclear human embryo to demonstrate the collection, and which is shown here is very efficient, and also no off-target was detected by current analysis, including whole genome sequence and deep sequence. And with this, I hope, under the strict ethical regulation and with the technical improvement, and also with the translation cooperation, base editor could be a precise strategy for collection of pathogenic mutation in an appropriate way in the future. And now I want to end the talk by giving my acknowledgement to all my lab persons, a very hard group, and also lots of my collaborators, and most of the embryo work are did by the professional team. Also my support agency here. And finally, I want to thank you all for your attention.
Thank you. So could I invite the other speakers to come on the stage? We will have now, as I mentioned before, a question and answer session for the 4 speakers. We can't make it very long, but there will be opportunity over lunch, in theory, to ask questions if you want. Okay, so first question, please.
I have a question for Dr. Amato. It's puzzled me that since the first S phase in the human embryo occurs when the pronuclei are still separated, that if a break occurs very early in the male pronucleus, that the broken chromosome will be attempted to be replicated before it ever sees the maternal chromosomes. So do you have an idea when during those first stages of embryogenesis that the male and the maternal and paternal chromosomes actually see each other?
Paula, can you just wait a sec? Please, can people in the room be quiet, because it's not very nice to the speakers if you're making a noise. So please be quiet.
Thank you for your question. So I'm not sure we understand exactly when the repair happens. We think the cut happens at the introduction of CRISPR, but the repair may actually happen later on. And I understand that the pronuclei are separate, but at some point they interact because, of course, you get recombination. That's how inheritance works. So our hypothesis is that the cut may occur early, but the repair may be delayed.
Yeah, it still seems sort of anomalous, and there isn't necessarily recombination after fertilization. The intentional recombinations occurring during meiosis. So it's puzzling.
Yeah, we're still studying it.
Anyone else want to comment on that? Or, can I have a quick question, which is sort of relevant, which is to both Maria and Paula. There's a lot of evidence that sperm DNA can be fragmented. And so, of course, it's hard to show that a sperm with fragmented DNA has gone on to give us a good embryo, but the proportion of fragmented sperm with fragmented DNA is very high. So that has to get repaired somehow. Is it understood how that mechanism works?
I don't think so, and I think that's a really excellent question, and I'm not sure I made this point clear enough in my talk, but I think up until these kinds of experiments, it wasn't a priority to really understand repair in the early embryos, but I think now we know that there are lots of questions that need to be addressed and that sort of thing about DNA damage in sperm. If it's just simply repairing nicks, that's not an issue, but if it's really complete fragmentation of the sperm genome, that would be something quite profound.
Question here then.
I have a question for Dr. Nayakan. I think you made a really compelling case for the need for using human embryos in your research, given the significant difference with mouse embryo models. But my question is, you talked about procuring human embryos for the research because of partnerships with surplus embryos left over from IVF cycles. And I was wondering if you could talk about some of the best practices in ensuring a ready supply of embryos for research, particularly given the promise of your work for reducing some of the inefficiencies in IVF cycles. The whole concept of surplus embryos may actually sort of fall to the wayside if your research is successful.
So if I understand it, your question is about what is the process of obtaining these surplus embryos and obtaining kind of informed consent.
Yeah, if you could give us a sense of best practices that you would recommend. Thank you.
Sure. So in the UK, we have a dual regulatory system such that there is a national research ethics committee. So the Health Research Authority regulates the research ethics committees, and that's at the national level. And then there's regional research ethics, but it's not at the level of isolated, institutions. So it's kind of a higher level than an IRB in effect. And then in addition to that, we have a national regulator specifically for human embryo research. That's called the Human Fertilization and Embryology Authority, and they license research. They also audit us and all other research kind of involving human embryos, and they also audit and perform license reviews of any clinical practice. So the process of actually going and obtaining informed consent involves approval from a research ethics in terms of the consent documents, and then in addition to that, approval of the project via the HFEA, and then within the HFEA, there are two separate approval committees. So it's quite involved, but but I think it's quite rigorous as well, which gives the public confidence. And then it's a matter of distributing those consent documents and patient information sheets and ensuring that the patients are informed. I hope
It
that
seems like
answers
the UK model is commendable. What about in a privatized setting like the US and China, where you have private...
I think that's a discussion for another time.
Thank you very much.
Sorry. Question here, please.
My name is Ishii. I want to ask Cassie, you showed, at the last slide, you showed chromosomal abnormality after embryonic editing. So, but a genome of human early embryo is very unstable.
Yeah.
And my question is, can you distinguish naturally genomic instability from the off-target effect?
This is a really good question.
Yeah.
So I think that numbers are too low to distinguish that, and it's an extremely important question. So the way that I would think about doing this is somehow statistically determining what's the probability of having these segmental losses and gains consistently at this OCT4 target locus in X number of cells and in X number of embryos compared to the segmental losses and gains that are observed in IVF embryos, irrespective of whether they've ever seen a CRISPR-Cas9 break. So I think that's an extremely important question, and it's something that what I'm hoping is, as a community of individuals working with early human embryos, because they're so rare to work with, perhaps we can have a dialogue about how we can exchange information and learn from each other, so we can increase the number increase the information we gain, but decrease the number of embryos that are used to gain that information.
Okay. Thank you. Last question, I think, and then...
Thank you, Robin. Kevin Davis from the CRISPR Journal. I wanted to follow up on that question for Kathy. Maybe the numbers are too low, but could you just give the frequency of the rearrangements that you observed? And is there something special about chromosome 6?
No, I don't think so. My suspicion is that this is probably occurring in other embryos, and it hasn't been looked at because, firstly, it's quite expensive to do whole genome sequencing at the single cell level. But I think this is the type of approach. I think we had a discussion yesterday about some of the consensus about perhaps introducing, what are the systems in place to evaluate potential off-target events. I would add to that potentially also having quite a rigorous evaluation of on-target editing as well, especially in a human embryo context, or any primary human cell, really.
Thanks.
Yeah.
Okay, I think we should probably call it today then. Obviously, you can ask questions of the speakers. Thank you.